discussed so many hormones or so many secretions of the various glands, that is endocrine glands, as well as the exocrine glands. Now, according to this topic, today's topic is the insulin hormone, insulin. Now, this insulin we will discuss under the heading as definition, under the heading of definition, nature of the hormones, organ responsible for the synthesis, is pancreas considered as a gland? First hormone that was isolated and synthesized by recombinant DNA technology. Now, one by one we will discuss this point. First of all, the definition, as it is written that it is anabolic hormone. Now, insulin is a hormone which is known as anabolic hormone because it helps in the synthesis of the carbohydrate, protein, and the lipids. So that's why we can say that it is an anabolic hormone. So other is that nature of the hormone. Nature of the hormone, that means it is a proteinaceous hormone, because you know that I told you that there are various classification of the hormones. Either they are amino acid derivative, or they are the steroids, or they are the proteinaceous. Proteinaceous means those hormones which are synthesized by the amino acid. So they are known as proteinaceous hormone. Now, the organ responsible for the synthesis. Now, the organ which is responsible for the synthesis is the pancreas. Now, pancreas, you know that it is that part of the body, it is that part of the gland which synthesizes the two hormones basically. One is known as glucagon and other is known as insulin. Now, glucagon and insulin can be synthesized by the pancreas. Now, question is this, that is pancreas considered as a gland? So, we can say that yes, pancreas can be considered as a gland because it secretes its secretion, which is endocrine secretion as well as exocrine secretion. Endocrine secretions means those secretions which comes under the heading of hormones, and they are directly poured into the blood circulation, for example, insulin and glucagon. Whereas the exocrine gland means that they secrete their secretion into the intestine or any other part of the body through the ducts. Now, pancreas secretes the secretion exocrinally, means that which are known as exocrine secretion. They are usually those secretions which enters into the intestine by a duct known as pancreatic duct. So, presence of this duct differentiate the endocrine and exocrine secretion. Now, the exocrine secretion of the pancreas is nothing but those secretions which entered into the small intestine through the pancreatic duct, and these secretions help in the digestion of the proteins, carbohydrate, and lipids. So that's why, due to their secretion, beside that there is secretin, there is pancreazymin, there is cholecystokinin. These are also the hormones, and they are known as exocrine secretion. They enters into the intestine and then they participate for the activation of various enzymes which participate in the process of digestion and the digestion of the different food components, right? Now, it is the first hormone that was isolated and synthesized by recombinant DNA technology. Now, recombinant DNA technology is that technology in which we can synthesize by the process of the genetic material, and this process takes place in the synthesis of various drugs, various hormones. So, insulin was the first hormone which is synthesized, which can be used as a drug, and it is synthesized by means of DNA technology. Now, coming to the chemistry, because we are going to discuss this hormone under the heading of biochemistry. So, the chemistry is very important of this hormone. Chemically, this hormone having the two chains, A chain and B chain. So, number of amino acids, because as I told you that insulin is a proteinaceous hormone, it is composed of amino acids. So, these amino acids are present in two chains, chain A and chain B. Chain A is having 21 amino acids and chain B is having 30 amino acids. Now, A chain and B chain, previously they are also known as alpha chain and beta chain. So, alpha and beta. So, A chain and B chain. Now, total number of amino acids present in the insulin is 51. The number of total amino acids present in 51. So, 51 amino acids are present in the insulin in A chain and B chain are responsible for the biochemical activity, Now these two chains, they are not in a floating condition, they join with each other by means of a bridge that is known as sulfur bridge. Now it has been observed that these two chains connected by disulfide bridges, and disulfide bridges, you know that they are present between the amino acid cysteine, cysteine of one chain to the cysteine of the other chain, that is disulfide bridges present between. Now these chains, there are 3 bridges are present. The most important are the 2 which connected the A chain and B chain. The number of amino acid A7 connected with the B7, that is the 7th amino acid present in the A chain and 7th amino acid present in the B chain. There is a connection by means of disulfide bridge. Second is the connection between the 20th amino acid of A chain and 19th amino acid of the B chain. Again there is disulfide bridge. Beside these 2 bridges, there is third kind of the bridge in the A chain is also present, which is among the amino acid, that is amino acid 6 and amino acid 11. That is known as the third chain, right? So it is the structure briefly. Now the gene which is responsible for the synthesis of insulin, it is usually located on the chromosome 11. Now chromosome 11, this gene also known as INS gene, means insulin gene. This gene is responsible for the synthesis of the insulin, and it is present on the chromosome 11. Now first there is now how this insulin is synthesized in the body. Insulin is synthesized in pre-pro-insulin form. Now you have known that I have discussed with you about the zymogen form. Zymogen forms are those forms which are formed when they are formed, they are present in an inactive form. By the removal of certain amino acid, they becomes in the active form. So similarly, insulin when synthesized, it is present in pre-pro-insulin form. The pre-pro-insulin is having 108 amino acid. 108 amino acid. Pre-insulin 100 amino acid. Pro-insulin, it is having 86 amino acid. The molecular weight is 11,500 Dalton, whereas pro-insulin having 86 amino acid, and these two are connected by means of before this, because still now up till this point the insulin is not present in an active form. It is present in an inactive form because there is a presence of an extra chain or extra peptide that is known as C-peptide. Now this C-peptide is that peptide which is responsible for the connection of the peptides, and this is having 31 amino acid. Now you can see in the figure that how this insulin synthesized and what is the pro-insulin, pre-pro-insulin and the C-peptide. Now this figure elaborate about this discussion. Now you can see here, this is the synthesis of insulin, that is pre-pro-insulin, 108 amino acid. Now this is the structure in different colors. This is the red color, black color, and this is the green color. This green color having the amino acids, 23 amino acids present in this from one point to this point, and this is known as ladder protein. This is known as ladder protein because it is responsible to give the strength to the structure of the protein. That's why it is known as ladder protein. Now beside this, there is formation of pro-insulin by the removal of certain amino acid. Then the remaining amino acid are 86 in number, and this is, now you can see here, the blue color, the red color and the black color. Now black color is nothing but the C-peptide, which I have just told you that it is having 31 amino acids. It is known as C-peptide, whereas this is the A peptide and B peptide, that is with red color and green color. Now A peptide, I have told you that it is having a self bridge that is known as sulfur-sulfur on the A chain, whereas two bridges are present between this A chain and B chain, and this is disulfide linkages. Right? Now again, coming to the point, that insulin, which is having a 51 amino acid. Now first of all, there is removal of the ladder protein, then there is removal of the C peptide, and then there is formation of the active form of the insulin. Now this is the active form of insulin. This is the A chain, this is the B chain, having 21 amino acid, having 30 amino acid. These are the two bridges, disulfide bridges, which are present with the semin and the 11th amino acid, 19 and 20th amino acid, and third chain present between the carbon number 6 and 11 of the A chain. So this is the active form of the insulin. This is the active form of the insulin. Now, again coming to the same slide, that is how we have discussed this. That is, now this is the, because of this we have discussed C peptide. Now that was the figure which shows the structure of insulin. Now where the insulin stored? The insulin stored in the granules of the Golgi apparatus. Now Golgi apparatus is that part of the cell or the cell organelle which have the sort of communication between the intracellulars, between the organelles of the intracellular. So this is also having the zinc ions. This is also having the zinc ion in their granules. Now, how this insulin is released from these granules? How this insulin is released from these granules? Now, these granules, that is the zinc, can be released by the process of exocytosis. Now again, I am going to repeat that pancreas, you know that pancreas is that part of the gland which having two parts of the cells, two kinds of the cells, A cell and B cell, and that is known as the cells of the Gland. A cell secretes the hormone glucagon, whereas B cell secretes the hormone known as insulin. So insulin is secreted by the beta cell of the or B cells of the pancreas, that is the Langerhans cells of the pancreas are responsible for the secretion of insulin, right? Now what is the normal amount of the secretion of insulin? Normally insulin is secreted 40 to 50 units per day. Now there are certain factors which are responsible for the activation or the release of insulin, and so there are certain factors which are responsible for the inhibition of the release of insulin. Now first, the factors stimulating insulin secretion. These are glucose itself, then amino acid, that is arginine leucine, particularly the arginine leucine, then GIT hormones, secretin, cholecystokinin and pancreasidin. These are the factors which are responsible for the stimulation of the insulin or for the release of insulin. And you know that insulin is released by the process of exocytosis. Because you know what is the process of exocytosis? Means that when there is combining of the membrane of the Golgi apparatus with the cell membrane, there is process of breakdown of the membrane takes place, and that is known as exocytosis, and there is release of the insulin. Now factors inhibiting is particularly the main factor which inhibits the release of the insulin is known as epinephrine. Now epinephrine, last time I have told you that it is a secretion of the medullary part of the adrenal gland. So this medullary part of adrenal gland secretes the hormone epinephrine, and this epinephrine is responsible for the process of glycogenolysis, whereas insulin is responsible for the process of glycogenesis, right? Secondly, it causes the lipolysis, whereas insulin helps in the synthesis of the lipids. Now that's why I've told you in my beginning of the lecture that insulin is an anabolic hormone, not the catabolic hormone. So epinephrine causes the glycogenolysis and lipolysis, so it is the catabolic hormone. Again, you know that these processes shows their mechanism of action through cyclic AMP, which we have discussed various times in our previous lectures, right? Now, how this glucose enters into the cell? How this glucose enters into the cell? You know that glucose enters into the cell. What is the role of insulin? To increase the utilization of glucose. To increase the utilization of glucose. So that's why glucose can be utilized by the body in the presence of insulin, by the body in the presence of insulin. Now again, how this glucose enters inside the cell. You know that if you remember one of my lecture in which I've told you in detail about the glucose transporters. There are various glucose transporters starting from glucose transporter 1 up to glucose transporter 5. Now among that, the most important is the glucose transporter 4. Glucose transporter 4 is responsible for is responsible to take the glucose from outside the cell, then keep them in them, and then this glucose enters inside the cell, and then this transporter pour out this glucose inside the cell, where this glucose is oxidized by means of various pathways which we have discussed under the heading of carbohydrate metabolism, that is by the process of glycolysis, by the process of hexose monophosphate shunt, by the process of citric acid cycle. All these are the processes for the oxidation of glucose in our body. Now you can see in this figure, this is the glucose transporter. These are the glucose transporter. They are present inside the cell. Previously they are present inside the cell. This is the plasma membrane. This is the glucose molecules. This is the glucose transporter. Now what is the role of insulin? When insulin comes, when glucose is there, insulin, there are certain factors, and I told you just now that glucose, one of that factor which causes the stimulation of the activity of the insulin, right? So insulin when activated, it combines with its specific receptor, and again I am repeating that the proteinaceous hormone, they are having their receptor on the cell surface level, on the cell membrane level. Now this is the insulin combined with the specific receptor, and then this receptor responsible to give a signal. There is transport, there is transport vesicles. So signal, when there is stimulation of the signals, there is communication between the glucose and the insulin and the glucose transporter which is present inside the cell. Glucose combined with the receptor, then that is the insulin combines with the receptor, there is activation of that receptor, and there is arising of a signal. A signal takes place that is known as translocation. So this process takes place, and there is this signal activate the glucose transporter that to go to the cell membrane immediately to take the glucose, and then after taking the glucose again they enters into the cell and they releases that glucose, and inside the cell there is oxidation of the glucose or the utilization of the glucose takes place. So we can say that signaling of this insulin receptor complex causes the process of translocation. That is, translocation means by means of the chromosome 11, which I just explained you. The chromosome 11, it is a gene that is responsible for the process of translocation, right? So due to this translocation, there is activation of the transporter, then it comes to the surface membrane, and then on the surface membrane they take the glucose, then they enter back into the interior of the cell. They releases this glucose inside the cell, whereas the enzymes are present which are responsible for the oxidation of glucose, Now, you know that this oxidation of glucose takes place inside the cell. Now, what is the mechanism? How the glucose enters inside the cell? It is not a simple process. It is not a simple diffusion. It is not as the membrane transporter or transportation. The entrance of, be careful about this point, the entrance of glucose from outside the cell to inside the cell against the concentration gradient, because it requires energy. All those processes which takes place against a concentration gradient means they require the energy. Now from where this energy comes? This energy comes from the ATP. Now again, you know that during the topic under the heading of metabolism of carbohydrate, I told you in detail about this transportation of glucose against a concentration gradient, and I have told you that glucose follows the same pathway for the entrance from outside the cell to inside the cell as the sodium ions, So concentration gradient, so adopted the same pathway as sodium, that is ATPase dependent mechanism. ATPase dependent mechanism. ATPase is that adenosine triphosphatase which act upon ATP to convert it into its component ADP, AMP or PI, that is inorganic phosphate. During the breakdown of these bonds, there is release of energy, and that energy is then utilized for the transportation of glucose from outside the cell to inside the cell. So this is the mechanism of